Hi everyone, today we are going to discuss about animal kingdom. We have already learned the animal kingdom in class 9, but class 11 we are going further more detail. Before going to that, we have to understand what you mean by an animal. Animals are actually the multicellular, you can see that they are multicellular and heterotrophic. It will depend on the other organism, right, for their food and all, and they doesn't have any cell wall, and chlorophyll is also not there. That means they cannot produce their food material. So animals are multicellular heterotrophic organism without cell wall and chlorophyll. Right. Now we have to understand that what are the general characteristics of an animal. Right. So here you can see that there are 11 phylum around, under the animal kingdom. What are they? You can see that that's actually the phylum Porifera. Right. So the sponges and all coming under here. Phylum Cnidaria or phylum Coelenterata, right. Another, the third phylum, that's actually the phylum Ctenophora, and the fourth phylum Platyhelminthes, the fifth phylum Ashhelminthes, or you can see that the Nematoda, right, and the sixth phylum Annelida, which is the phylum of earthworm and all, and the seventh phylum, which is the largest phylum Arthropoda, phylum Mollusca, phylum Echinodermata. These all are actually non-chordates, right, and you can see that the phylum Hemichordata and lastly the Chordata. These all phylum with some example and features we will go in detail. Before going to that details, we have to understand what are the general characteristics of an animal kingdom and what are the basis of classification. So here you can see that how we have classified the organisms in animal kingdom. The animal kingdom, the main basis of classification, these are levels of organization, that means whether they are cellular level, tissue level, organ level or organ system level, like that. Then we have checked that body symmetry, whether they are bilateral symmetric, asymmetric or radially symmetric. Then they have checked about that the embryonic layer, the germinal layer such as ectoderm, endoderm and mesoderm. After that they even taken as a basis the coelom, the body cavity, the presence of coelom. Cenomate hai, ascoelomate hai or pseudocoelomate hai, wo bhi dekh liya hai. After that the segmentation is there, that means they are metamerically segmented. Metamerism is there, present or not, right. So that's the also taken as a basis of classification. Then at last they have taken the presence of notochord and the absence of notochord, that was also a basis of classification, right. So we will go one by one in detail. Here you can see that the basis of classification, the first one, the level of organization, right. The level of organization actually you can categorize into different types. First one is the cellular level, in which all the organism which is having unicellular or multicellular but it doesn't form the tissue, that is that form, cellular level. Then coming the tissue level, that means there will be those who are performing the same function and similar type of cells together will form the tissue and they will do the specific works, right. So there in some organism the tissue level of organization you can see that. In another you can see that the tissue again modified to form some organs, so that's actually organ or that organ level of that organization we are telling. Then you can see that the final organ system or the organism level, right. So here also you can see this, I have given all these things. The cellular level of organization, that's actually mostly common in the case of Porifera, the sponges and all, right. So that means the cells are aggregated as loose cells aggregates. You can see that arranged, the cells are arranged as loose cell aggregates. That means it's multicellular but it doesn't form the tissue level. It's simply a group of mass of cells together and they are performing their corresponding function, right. Now the second level, that's actually the tissue level of organization, in which you can see that the similar cells will arranged or they will perform some particular function, that's actually called the tissue, right. They will arranged into to form the tissues. Examples are the Cnidarians and the Ctenophora, right. Both group are coming under tissue level of organization. The next level you can see that the organ level of organization, mostly common in the case of higher organisms, right. You can see that organ will be there, special organs will be present. That's actually mainly in the case of higher animals after Platyhelminthes to chordates, you can see that, right. and sorry, the picture is actually wrong here. You can see that the organ system level, right, that means above that, that means chordate, all organisms, you can see that, including animals, right, all the animals and all, they all are having organ system level. That means there will be a reproductive organ, excretory. In some of that actually arthropoda group also, insects, it's not actually completely wrong. You can see that in the case of that cockroach and all, you can see that the organ system level also, cockroach which is having that the reproductive system, excretory system, that everything is present there. So organ system level in higher animals, right. So even you can say that some of that chordate, that arthropoda also, they are having some level of organ system is there, but the highest level of organ system level, right, organs are actually associated with the organ system. So we can say that we cannot actually make a clear differentiation between organ level and organ system. It can, the organs are formed, then it can form the organ systems also, right. So these, each system will perform a special physiological function. Like that, excretory system, they will have excretion in function. So respiratory system, which is helping for the respiration altogether. Circulatory system for the passage and that circulation, transport, you can see that. So these are mostly commonly seen in higher animals, right. Now you can see that the level of organization in which system, you can see that along with the level of organization, you have to actually understand there are some other things are there, like digestive system, right. There are two types of digestive system most commonly, you can see that, that's actually incomplete digestive system and complete, right. So organ system of different animals show different complexities, and for that very important or very good example is our digestive system, right. In the case of, in our case, in human being, you can see that mouth is that actually the organ which is actually for the intake of food, and through anus the undigested food will be released out, right. So that means there are two openings here, right. So mouth and anus, then we will say that it is a complete digestive system. But in the case of hydra and planaria, all these organisms, you can see that there is a single opening through which the food is taken in, that will be absorbed. That means they have a special body cavity and all, that will be digested and absorbed here. After that, the waste material will also removed out through the same opening, right. So you can see that. So that's why it is called actually incomplete digestive system. That means there is only single opening for which is act as the both mouth and anus, that for intake of food and also excretion of that waste material. So there is only one opening is there, right. Complete digestive system, I told you that in the case of human being and all, you can easily understand there will be two opening, the mouth for intake and the anus for that removal of the excretory part, right. Now circulatory system also the same way, you can divide into two part, which is the open circulatory system and the closed circulatory system. What is the specialty here? In the case of open circulatory system, there is no blood vessels present here. There will be a very large, what will you say, cavity, which is called the hemocoel, right. Hemocoel, we can see that hemocoel, in which the blood will be filled here. All the organism, that all the organs will be immersed in this the cavity, and direct exchange, diffusion of that what will say oxygen and all other things will happen here, right. But in the case of closed circulatory system, it is actually, the blood is actually flowing through the blood vessels. The presence of blood vessel is very important in the case of closed circulatory system, and the blood is actually not at all coming out of that blood vessels, it's actually entirely passing through the blood vessels, you can see that. And it will open in some spaces only, the heart and all that. Then there will be exchange through the capillaries, there will be exchange of gases and all. So here one thing you have to understand, in the case of circulatory system open, there will be no blood vessels, there is rather than hemocoel is present, in which all organs will be immersed, and you can see that there will be direct exchange of materials will happen. In the case of closed circulatory system, there is actually the presence of blood vessels is there. Now the second type of second basis of classification, which is actually the symmetry, right. What do you mean by symmetry? It is actually the arrangement of similar body parts in two sides of the main axis, that's called the symmetry, right. Based on the symmetry, we can categorize the animals into two types mainly, asymmetrical and symmetrical. Asymmetrical means the body cannot be divided into two equal parts. You can see here in this picture, this body is actually, you can divide into two equal parts. What about here? here there is a symmetry, that means two equal part is there. Here you can see that it's not equal, it's different, here also it's different. And here you can see that if it is a sponge, right, that's actually हमारा Nidarians. Nidarian की case में क्या है, उधर it is not following any symmetry, it can be different shape, it can be, so that's why it is not at all following any symmetry, that means it is asymmetrical in nature. But in the case of beetle or anything, you can see that it's actually following the symmetry. You will get the equal halves when you are dividing, right? So, and what is that symmetry, that we will discuss, but simply you have to understand what you mean by symmetrical and asymmetrical. The body cannot be divided into two equal halves, then they are told that na two or what, that means equal half में divide ही नहीं कर सकती, तो asymmetrical. Mostly poriferans, कुछ snails वगैरह asymmetrical है. What about in the case of other organisms, you can see that mostly they are actually symmetrical, that means it can be divided into equal parts, right? Here the symmetrical, then there are two types of symmetry is there. One is the radial symmetry and other one is the bilateral symmetry. We will go and discuss in detail. Here you can see that this is an example of radial symmetry. Radial symmetry is actually the body can be divided into two equal halves where any vertical plane or along the which must be passes through the central axis. That's only that importance. If you are dividing, this is the sea anemone, if you are dividing that what will you say, that the entire organism, right, you can see that if you are any axis which must be passing through the central axis, you will get the two equal halves. Then we will say that they will allow that obey the radial symmetry, right? So you can see that this mostly some of the poriferans, not all, most of the poriferans are actually asymmetrical and some are actually obeying that. Some poriferans, some Nidarians or the Coelenterata, some Ctenophorus, that's a jellyfish category, and echinoderms, that's actually here also one thing you have to remember. Echinoderms, that's actually the category of that starfish, right? When they are in that what will I say, that in that larval stage, this echinodermata will show that radial symmetry, bilateral symmetry, and when they will become that adult, they will follow the radial symmetry. You can see that, that means starfish and all, you can see that they will follow the radial symmetry, but when they are in the larval stage, they can even form that bilateral symmetry, right? So adult echinodermata will not show the bilateral symmetry, but the larval stage and all other stage, they can follow the bilateral symmetry. Adult will show only the radial symmetry. That means if you are cutting the organism into into two halves, right, which the axis must be passed through the central axis, then you will get the equal halves. Then we will name it as they are actually following the radial symmetry, right? And what you mean by bilateral symmetry? Bilateral symmetry, you can see here, it's actually if the body can be divided into two equal halves, that's actually we are telling the right half and left half, when it will be cut through only one plane. If you are cutting like this, you will not get the equal halves. You must be cut like this. What will you say, that it must be cut like this, then only you will get the equal halves. That means only one plane you are getting two equal halves. They are called bilateral symmetric. Mostly Platyhelminthes to Chordata, all except adult echinodermata, all are following that bilateral symmetry. The lower organisms mostly following the radial symmetry. Now, what are the germinal layers you have to understand here? The main germinal layers means that actually what will I say, that embryonic layer, we can say that, right? So these are the layers of embryo from which all the body organs are forming, right? So based on the number of germ layers, how many germ layers are present in that organism, we can divide the entire animals into diploblastic and triploblastic. Diploblastic means they will have only two type of germ layers, which is the ectoderm and the endoderm. The innermost one, that's actually endo, and the outermost one ecto. Triploblastic animal means they will have ectoderm, the outermost one, and they will have endoderm, the innermost, and there will be a middle layer which is called the mesoderm. Then it will be a triploblastic organism, right? So here you can see that the entire body organs are forming from all these three layers. You can see that the ectoderm, the endoderm, and the mesoglia or the mesoderm cells, you can see here, which is here, right? Now According to the presence, I told you they are diploblastic and triploblastic. Diploblast means only two germ layers are present, the ectoderm and the endoderm, right? That's outer ectoderm and the inner endoderm will be there, and you can see that there will be a non-living layer in between them. You can see that they are called mesoglia in diploblastic organisms, right? So you can see that the jelly like layer which is called the mesoglia, right? Mostly these type of diploblastic organisms are in the lower level. That's actually the example of sponges and the jellyfish category, that's Cnidaria and Ctenophora. And the higher animals, mostly you can see that they are triploblastic organism. They have ectoderm, endoderm and even mesoderms also present in between them, right? So here there will be no jelly like structure in between the ecto and endoderm, that will be represented by the mesoderm, right? So if three germ layers are present all together, then they are called triploblastic organism. Platyhelminthes to chordata, all phylum are triploblastic, right? So that's why I told you the lower groups are diploblastic, and after that platyhelminthes, you can see that all are triploblastic in nature. And you can see that there will be a space in between the body wall and the digestive cavity, that's called the coelom, right? If the coelom is absent, then if the coelom is absent, then these organisms are called as coelomate. That means there will be no coelom present in this, right? So you can see that the spaces between the body wall and the digestive cavity, right, is filled with matrix, right? You can see that there is no coelom is present at all. You can see that this is ectoderm and this is the mesoderm and the endoderm, and you can see the digestive cavity is there. So there is actually only filled with the matrix. There is no coelom cavity, body cavity is not at all there, right? So you can see that that's actually called the coelomate. That means no coelom present here. Mostly examples are the porifera and the platyhelminthes, right? And you can see that porifera, Cnidaria and platyhelminthes. After platyhelminthes, you can see that there is a category that's actually the helminthes or nematoda. Nematoda is the only group which will show the pseudocoelom. Nematoda. Nema means thread like, helminthes otherwise known as the nematoda, right? Here what is actually, you can see the ectoderm is there, mesoderm is also present, and you can see the endoderm. But this mesoderm is not at all a continuous layer. It's actually, you can see that it's scattered pouch like structure. Mesoderm is actually scattered pouches. You can see that 1, 2, 3, 4, right? In between you can see that there will be a body cavity which is actually not lined by the mesoderm. Normally this body cavity, the coelom, will be lined by the mesoderm, but here the mesoderm is present as the pouches, scattered pouches. So there is no lining around the body cavity. So that's why it is called false coelom or pseudocoelomate, right? And if actually, so you can see that according to the coelom present, right, body cavity, we have discussed that the organisms into coelomate, right? And you can see that pseudocoelomate, and we can even divide it in them into coelomate. That means whether they are having the true coelom or not. That means you can see the ectoderm will be there, mesoderm will be there, and inner endoderm will be there. In between them you can see that there will be a cavity, right, which is the coelomic cavity, which is actually arises from the mesoderm, right? And this coelom will be lined by the peritoneal layer, right? Or inside that coelom you can see that there will be coelomic fluid also, right? So this true coelom, you can see all that after helminthes, right? You can see that all the phylum till chordata, that means Annelida to chordata, all the groups are having the true coelom. So once again we can see that porifera and Cnidaria and platyhelminthes, you can see that they are coelomate. Once it will become helminther or the nematoda, you can see that that's actually the nematoda, right? Nematoda, this is having only one group which is having pseudocoelom. After that, which is having the true coelom, that's actually Annelida to chordata, all phylum having the true coelom. If you are checking the coelomic cavity, whether the body cavity is present, that's I already told you as coelomate, pseudocoelomate and coelomate. You can see that what you mean by this. The coelom, it's actually the body cavity lined by the mesoderm. It must be mesodermically originated, right? and you can see that this is actually in between the body wall and the gut wall. The body wall, the outermost, and the gut wall, in between them you can see the coelom cavity is there. This is a cavity. Based on the nature of the coelom, the animals can be divided into three categories: ascoelomate, that means without coelom; pseudo-coelomate, that means it will look like coelom but it's not actual coelom, true coelom, and they are the coelomate. Right. All the other things you can see that all the body organs actually originated from the ectoderm, endoderm, and the mesoderm. Now the next basis of classification, that's actually the metamerism, or we can say that they are the segmentation. Whether the presence of segment, right. So it's the phenomenon in which the body or organ is externally or internally divided into repeated segments. You can say that that's actually the metamerism, right. So here this is an example of that Annelida, that's actually earthworm. You can see that there is actually segments in between them, right. And this is one of the example of very famous example, Nereis, which is an aquatic Annelida, in which also you can see that there will be segment present in their body. And you can see that next basis of classification, that's actually the presence of notochord. Here you can see that the notochord is actually structure, the embryonic structure, this that now supporting structure which is present at the time of that embryonic development, right. You can see that this is mesodermically derived, that's actually this notochord is actually forming from the mesoderm, and this is a supporting rod which is formed dorsal side, that means in the back side you can see the dorsal side at the time of embryonic development in the animals, right. In some animals this notochord will be present throughout their life cycle, right, or it can be persist there, right. If the notochord is present, then they are called chordates. If the notochord is absent, they all are included in non-chordates, right. So you can see that there will be norsal. If it is a chordata, then they have some features. They will have notochord. There will be other dorsoventral hollow nerve cord. You can see that pharyngeal slits will be there, right. All these things you can see that, right. So here at a comparative analysis or a short glance, you can see that all the phylum, the level Porifera, which is cellular level, Cnidaria or the Cnidaria and the Ctenophora, Cnidaria otherwise known as Coelenterata, right. These two categories are tissue level organization. Platyhelminthes, you can see that organ or even organ system also you can see that, as helminthes that organ system, Annelida to chordata, all that organ system level, right. And here you can see the germ layers. In Porifera, this is only aggregates of cell. We cannot even classify it into germ layer or anything. Cnidaria, they are diploblastic, and Platyhelminthes onwards, all other organisms, they all are triploblastic. If it is triploblastic, mostly they will be bilateral symmetry. You can easily understand that, easily remember, right. And before that you can see that the Porifera, which can be asymmetric or sometimes it can be radially symmetric. Cnidaria and Ctenophora, they are actually the radial symmetric. By any axis you will get the equal halves. And after that, if it is triploblastic, then they must be bilateral symmetry, except Echinodermata, that will be in the starfish and all, which is bilateral symmetric in the larval form. When it will become adult, then that will become the radially symmetric. That's only the exception in between them, right. And here you can see that the lower organisms all are Porifera, Cnidaria, Ctenophora and Platyhelminthes. They are as Cenomate, that doesn't have any coelom. Only one group is having pseudo-coelom, that's actually the ash helminthes. After that, all the members are coelomate. So here at a glance we have summarized the basis of classification of animal kingdom. I hope you have a clear understanding of these things. Thank you.